thank you for attending our webinar. We're excited to explore the image segmentation to scaffold registration pipeline with you today. This will be demonstrated through a two-part webinar series. In part one of the series, we will review image segmentation portion of the pipeline. We encourage you to attend part two on January 26th, where SPARC MAP-Core will show you how image segmentation data is used to derive finite element organ scaffolds. For those of you who do not know us, I'm Aiden Sullivan.
And I'm Macy Heel. We're part of the SPARC team here at MBF Bioscience, which is a subgroup of MAP-Core. We have a few other SPARC MAP-Core members on the line today, including MBF Bioscience's Scientific Director, Dr. Susan Tappan, as well as Dr. David Nickerson and his team at the Auckland Bioengineering Institute. We'll all be available to answer questions at the end of the webinar. We encourage SPARC researchers to also reach out to the entire SPARC MAP-Core team on Slack. We have a channel there, #MapCore-support. So please communicate with us through that channel at any time.
We have a lot of exciting things to discuss today, and through our practice sessions, we've run really close to our 30-minute limit. So we just like to let you know that we're happy to stay for an extra 10 minutes to answer a few questions at the end of our presentation, but please don't feel obligated to stay for this portion if you have to run. We completely understand. We encourage you to ask questions throughout the webinar. You can submit a question simply by clicking the orange arrow button to open the GoToWebinar control panel. Find the question panel and type the question in the box. Then click Send. If desired, you can undock the question box from the panel for a better view of the questions and answers. We want to assure those of you who are here and are not part of the SPARC project that this webinar series is not limited to SPARC researchers. Though this presentation will discuss a SPARC-specific application of MBF Bioscience software, the tools we've developed for SPARC will soon be available to all users. In our demonstration, we will show you how to leverage NeuroLucida 360's toolset to produce accurate, quantifiable, and fair digital reconstructions of microscopy image data. The SPARC acronym stands for Stimulating Peripheral Activity to Relieve Conditions, and it is an NIH Common Fund program that aims to accelerate the development of therapeutic devices that modulate electrical activity and nerves to improve organ function.
The SPARC program funds researchers all over the world to create anatomical and functional data sets specific to neural circuitry, to create new tools and technologies to modulate and measure nerve-organ interactions, and to build a publicly available resource portal to share all this data so it can be remixed, visualized, and computed. The diverse experimental data generated by SPARC investigators is highly curated and made available for exploration and download on the SPARC portal. In addition to the source files, the SPARC portal also hosts dynamic organ maps, computational models, and simulations. Examples of the 3D organ maps, which we'll call scaffolds here and throughout the presentation, can be seen here on the right. They are finite element models of specific organs that can be transformed to fit your subject. Neural pathway segmentation can then be mapped to their computed location on a generic or on an individualized scaffold for comparison within and across your studies.
4 core groups, MAP-Core, SIM-Core, DAT-Core, and K-Core, make up SPARC's Data Resource Center, and together they drive development of the SPARC portal. The Auckland Bioengineering Institute, or ABI, and MBF Bioscience are MAP-Core. The core focused on making 3D and 2D maps of the autonomic nervous system's connectivity. Today we'll be showing you how experimental data in the form of microscopy image segmentation can be ingested in the MAP-Core pipeline to enable dynamic exploration of anatomy and function across a range of species.
So how can you transform your microscopy image data into a scaffold visualization on the portal? The first step is segmenting that image data to bring it into a common coordinate framework for MAP-Core engineers to work their magic.
Right, Macy. SPARC investigators from around the globe are generating biological research data for the SPARC portal. In this pipeline, researchers have access to tools to perform fair image segmentation to prepare their data themselves before it is passed over to the ABI Scaffold team for registration. The fair segmentation portion of the pipeline includes these 4 main steps. First, researchers can access MBF Bioscience software for segmentation of their primary microscopy image data contained in their dataset. Besides segmenting the neuronal connectivity of your organ of interest, it is important to put the microscopy image data into context, adding metadata about your organ sample, about where your organ sample is derived from, the age and sex of the sample will help others understand your data. Whether you've imaged an entire kidney with a light sheet microscope or a small section of the colon with a confocal, you must add anatomical fiducial information in order to register this data to the 3D organ scaffolds. You need to add metadata to the file that answers the questions, what is contained in my sample and where did my sample come from? Adding fiducial annotations with contours or markers will allow MAP-Core to more accurately fit your data into the common coordinate framework of the 3D organ models before it is fit to your specific subject. Segmentation data that is produced will be added to the experimental dataset and made available to ABI for scaffold registration. An in-depth look of this part of the process will be presented in part 2 of the webinar series. For now, let's dive into the segmentation aspect first.
As I'm sure many of you will agree, microscopy image data on its own is beautiful and fascinating and gives us a window into extraordinarily complex biological systems. Capturing cellular features and connectivity in a digital format to derive morphometric conclusions accelerates discovery by allowing us to quantify what we see. Digital representations of neuroanatomical features captured underneath a microscope can be used for downstream analytics, mapping, and computational modeling. And the segmentation allows you to individualize those structures and associate whole cells or whole populations with functional data, such as ePhys recordings or RNA-seq data. Also, MBF Bioscience segmentation protocols and applications, we can help you ensure that your segmentation data is fair. Hayden, do you want to remind us what FAIR is?
Sure thing. FAIR is an acronym that stands for Findable, Accessible, Interoperable, and Reusable.
To be findable, data must have rich metadata with explicit data IDs that are searchable in a registry. We make this happen at MBF by connecting our software to SciCrunch, a data and resource infrastructure. We use SciCrunch's terminology portal to provide SPARC researchers with a dynamic, organ-specific lexicon of biomedical terms to use while they're segmenting their image data. To be accessible, data must be retrievable by identifiers and have open protocols of how to understand the data. The segmentation data produced in MBF software automatically has software-specific identifiers that are registered in SciCrunch, and an open specification of the file format is available to the public. All segmentation files generated for a SPARC experiment are published by the researcher with the dataset on the SPARC portal, available for anyone to access and download. Our file format specification, which we'll discuss later on in the presentation, also enhances the interoperability of MBF-generated segmentation files. To be interoperable, knowledge must also be represented with FAIR approved terminology. In the cases we present today, we will discuss how the use of SciCrunch provided anatomy terms will allow MAP-Core at ABI to interoperate with our segmentation files. And finally, to be reusable, datasets must have sustainable metadata that complies with SPARC data sharing standards.
Before we dive into the FAIR image segmentation pipeline with a few use cases, we want to underscore that the variety of image data that can be ingested into the image segmentation pipeline. MBF Bioscience software supports a variety of image formats for microscopy vendors like Zeiss, Olympus, Nikon, Andor, Leica, and more. Though our first use case will demonstrate the pipeline from a whole organ perspective, it is not necessary that your data fit this mold. Whether you're segmenting a small portion of the left lobe of the mouse lung or a whole cap bladder, We can help you prepare your data for integration into the 2D and 3D maps produced by SPARC MAP-Core.
All right, let's take a look at our first use case. This experiment was performed by teams at Thomas Jefferson University and the University of Central Florida. Efforts were spearheaded by Drs. Jack Chang, James Schwaber, and Raj Vajapalli. I see a few of you here today. Thanks for joining us, and thanks for letting us show off this wonderful data set. So their group used the SPARC segmentation and scaffolding pipeline to help them answer the question, how does the intrinsic cardiac nervous system differ between individuals and across sexes? Intact hearts from male and female rats were excised, sectioned, and stained with crystal violet. The whole organs were imaged on a knife-edge scanning microscope and compiled into a 3D image stack for segmentation. Researchers used tissue mapper software from MBF Bioscience to delineate anatomical regions of the heart, such as the endothelium of the right ventricle, the aorta, pulmonary arteries and veins, and so on. They then identified and counted intrinsic cardiac neurons with a yellow marker seen here in the middle there. Individual segmentation files of select subjects were then registered to their own custom-fit scaffolds using unique fiducial points and the detailed structural segmentations. To allow comparison of ICN populations from a male and a female rat, ICN coordinates were then mathematically projected onto a generic rat heart scaffold. Details of this part of the pipeline will be demonstrated in part 2, but for now, let's take a look at how the team prepared their data files for integration into these visualizations. So we're gonna open up Neuralucida 360 here and show how some of that was done within the software by the researchers. So we've opened an image in the software just by dragging and dropping the image stack directly into the program window, and a welcome window appears where a researcher can indicate metadata about the subject that they're annotating. So here on the left, we'll select rat, give it a subject ID, and then if you know, you can indicate the sex and age of the subject as well. And then on the right-hand side, this is where we're going to integrate with the SciCrunch database services. We want to pull down a list of anatomical terms to use while we're segmenting. Here we'll pick the rat heart and a species independent list so we can get all the heart terms that we possibly want to use. This is a quick fly through of the image stack going in the Z direction, just plane by plane here. The image was cut at microns between image planes, and you can see the general shape of the heart. It's tilted on its side, but what I'll do is we'll open the 3D window so we can see the whole heart in a 3D environment. And what the researchers did is they annotated the anatomical structures of interest plane by plane at a certain section interval. So we're going to use the serial section manager on the side here to navigate somewhere in the middle of the heart, and I can show you how they contoured. So when we hit the contour selection button, you'll see that a list of terms, it's a big long list of cardiac terms, appear on the left-hand side. These are from SciCrunch, and they have a unique identifier that's written to the contour element once you trace something. We can search for a region of interest, the right coronary artery, for example, select it, and then click to detect the shape that you're interested in delineating. The researchers here also marked the intrinsic cardiac nervous system cells by using a marker. And so they zoomed in on regions of the ganglia here and clicked to place those markers where those cells were located. They repeated this process across multiple sections so that the whole 3D heart could be visualized, as seen here. But an important part of getting all this information registered to a scaffold is identifying unique points, so unique fiducial points, as Aidan mentioned earlier in the presentation. So I'm going to give you an example of how to do that using the SciCrunch terminology services. We'll use the marker bar here on the left, and we'll open a marker, and we want to rename it. So eventually we'll get there. We'll right-click on a marker and show the marker names of each of the markers. Then we'll choose one and right-click to say rename markers. And you'll see a similar list has appeared on the screen that looks like the list where the contours. It's the same list. We're pulling it from SciCrunch directly. In this case, we're going to click on the apex of the heart. The researchers determined where this point is, so I'm kind of cheating here, and we'll just skip down to the section of where that apex is. Placing markers and contours can be done in the 3D environment and in the 2D environment. In this case, they use the 2D environment to delineate these regions. And here I'll just click and place the apex. So now that we have a segmentation, we want to look at how the fiducial points match up to what the generic scaffold fiducial points look like. Again, to help this process of getting the data from segmentation into the scaffold pipeline. So I'm just going to turn down the image transparency here and the contours so we can see those markers. I have the yellow ICN markers, but then I also have 3 other fiducial points. And I'm going to bring in the generic rat heart scaffold by clicking on the scaffold icon there, pulling in that VTK file, and you'll see we have 2 viewports now, one on the left-hand side that has this generic rat heart scaffold with some fiducials identified already, and then we have on the right-hand side our segmentation file. If the names in the rat heart scaffold match the names that you've named your fiducial points, you'll see that the color of the fiducial marks in this scaffold file on the left-hand viewport will match the colors that we have on our 3, the orange, pink, and the blue square there as well. This process can be repeated across your whole experiment, and that is what the team at TJU and UCF did. They did this across 7 male and female rat hearts, and their data set is available on the data portal as well, and I encourage you all to go check it out. Here's this of those 7 samples. So.
Thanks for that conceptual use case, Macy, demonstrating whole organ segmentation. Let's take a look at another example. In this demonstration, we're going to explore one example of how a SPARC researcher has utilized the multi-resolution image segmentation strategies MBF Bioscience software can provide. This is a segmentation workflow that the John Tompkins lab at UCLA has used to understand the structure-function relationship of cells within the stellate ganglion. Their lab has backfilled cells within the stellate ganglion and recorded electrophysiological data for that cell. Next, they imaged the cells using an epifluorescent microscope at 40x to easily distinguish their neuronal pathways. They segmented the neuronal structures in high-resolution image using NeuroLucida 360's automatic tracing mode until the relevant structures of the image were represented. The Tompkins lab additionally imaged the whole stellate ganglion in 3D at 10x magnification, which is shown here. To put the backfilled cells into perspective, the high-resolution neuronal reconstructions were registered with the 10x image data for the entire stellate by moving the tracing in the X, Y, and Z directions until it is aligned with the low-resolution stellate image. You can see an example of the tracings being moved in the X and Y direction here. And here's an even more zoomed-in view of that. Using the FAIR vocabularies in NeuroLucida 360, the stellate structure from the low-resolution image was contoured on a series of 2D plans for a set interval. You can see this process happening. I'm selecting the cervical ganglion, a synonym for the stellate ganglion. and contouring the outer edge of the stellate ganglion in the 2D serial sections. A surface can be created with these contours in the 3D window to represent the volume of the whole ganglion in the data file. Additional structures, such as the cardiac nerve, are segmented to provide fiducial landmarks. You can visualize the axons of the yellow and pink cells innervating the cardiac nerve here. Next, the lab labeled the cells with their electrophysiological recording IDs and linked the physiological data with the morphological data to analyze the structure-function relationships of these cells. The data file can be used to enrich the generic stellate scaffold with fiducial information, and the morphological and physiological data can be registered to the stellate scaffold for visualization on the SPARC data portal. Using the scaffold fiducial matching tool, we can make sure that all appropriate fiducials have been added to the segmentation file to allow for the most accurate mathematical computation to determine the fitness during the scaffold registration process. Here, I'm circling a fiducial marker that has not been mapped yet. Once I add my marker in representing the junction between the stellate ganglion and the 8th cervical spinal nerve, you can see the marker color on the segmentation now matches the scaffold viewer. With the registration of this data to the stellate scaffold, the Tompkins lab was able to use the scaffold to visualize a heat map representing the locations of each cell body whose axons innervate a specific nerve exiting the ganglion. In this case, that's shown on the screen here, the scaffold is showing the innervation of the inferior cardiac nerve located at the top right of the stellate ganglion. We can clearly see that these cell bodies are localized within the same region of the stellate. This novel discovery further contributes to our understanding of the structure-function relationship of the cells within the stellate ganglion. Let's take a look at another use case and see what happens if your microscopy image data does not capture a whole organ or structure, like our past two examples. To demonstrate how microscopy samples of a small subsection of an organ can be registered to a scaffold, let's explore the segmentation workflow of Li-Shin Wang from the Dr. Tache's lab at UCLA. They use this workflow for reconstructing a set of 3D confocal images of the enteric nerve plexus in the mouse colon. These images, labeled via multicolor adeno-associated virus, were reconstructed using Neurolucida 360 to understand the cellular connectivity within the colon. Thank you very much for allowing us to use these stunning images and segmentation for our demonstration, Li-Shin. The segmentation techniques this lab has used for reconstructing their image data and preparing it for registration to the colon scaffold include automatic soma detection and fiber tracing. And you can see an example of this in the screenshot of column 2 on the slides here. To prepare this section for the registration of the scaffold, the myenteric nerve plexus of the mouse colon registration, the team utilized the following techniques to provide context for the scaffold registration team. The first step was naming the nerve fibers using the FAIR ontology list as the myenteric nerve plexus, describing this data with a unique name and identifier to answer the question, what is contained within this sample? They also used the sets feature in Neurolucida 360 to highlight important connections representing neuron communications. Each connection was labeled with the unique set name and included in the data elements, included unique data elements such as somas and trees. Lastly, the orientation and location of the sample was communicated directly to the ABI scaffold team to indicate where the sample should be placed on the colon scaffold. You can see the final results of this on the last panel of the slides here, demonstrating the segmentation registered to the colon scaffold. This segmentation data is not only compatible with the organ scaffolds, but it also allows for quantitative analysis via Neurolucida Explorer. Here you can see a summary of the analyses available in Neurolucida Explorer. This lab, in particular, analyzed their segmentation data to determine the major and minor diameters and ratios of the cell bodies. Check out the results in Lesion's full manuscript. The DOI is linked on this slide. Okay, so you saw how to obtain qualitative analysis from Neurolucida Explorer, but how can you use these data files if you don't have access to that software? We have an exciting announcement to make. MBF Bioscience has fully documented their file format for open use. It details each element of the MBF Bioscience XML data file in an effort to encourage the reuse of these rich data files for alternative analysis or reproduction of derived conclusions. This specification is used by the MAP-Core team for registering image segmentation data to the organ scaffolds. We hope this valuable resource will help your team too. Check out the file specification at MBF Bioscience/file specification. You can also ask us any questions you have about the file spec using our web forum. The link is posted on this slide here. And lastly, we published a preprint version of our related manuscript on bioarchives. Take a look and discover what makes this file format for neural anatomical structure modeling so impactful.
Thanks, Aiden. That was a great demonstration. Everybody, here's a recap of some of the tools that are in MBF software like Neurolucida 360 that we hope can help you to prepare your segmentation data for scaffold integration and help you to generate morphometric data to answer your individual experimental questions. We've added a lot of functionality to our software since we first got involved in the SPARC project. The scaffold viewer for fiducial matching, an API connection to the SciCrunch infrastructure for fair data production, and even ways to programmatically verify that each annotation has a unique identifier, whether you've created the segmentation file months ago or yesterday, you can programmatically check those. We've also amended the software to better handle really large image data because data sizes keep getting bigger every year. We've made improvements to our 3D environment window to help you visualize and process your data faster and more intuitively. We're also brewing up some new tools this year, including a tool that will detect the orientation of muscle fibers in your organ of interest. We're also hoping to build a bridge to MAP-Core's scaffold making software, which they will likely demonstrate in part 2 of the series here. So researchers can do everything from image segmentation to scaffold fitting, all from within Neurolucida 360.
If you're part of the SPARC investigator team and have not yet gotten a license for MBF Bioscience software courtesy of the NIH, please email us at sparc@mbfbioscience.com to get started today. Macy and I will also provide you with training and in-depth protocols to help you with your SPARC mapping efforts.
We're going to pass the mic all the way down to New Zealand for part 2 of the webinar on January 26th. Part 2 will cover how MAP-Core uses the image segmentation data to build subject and species-specific finite element organ scaffolds using their scaffold maker software. A link to register will be available soon and broadcasted on the SPARC announcements channel on Slack. On behalf of MAP-Core, we look forward to seeing you there.
Well, it looks like we've used up almost all of our allotted time. We want to thank you all for attending our webinar today. Macy and I are available to stay for about 10 more minutes to answer a few of your questions. Again, if you need to head out, please feel free to do so. We understand everyone's schedule is busy. We want to remind you that Susan Tappan and the ABI team in New Zealand are on the line to answer your questions as well. Thank you again for attending, but let's get started with some of those questions.
Looks like we don't have many, so we may be able to not take too much of everybody's time today. We really appreciate you all joining us. And if questions arise later and you're curious, again, Slack, email, let us know how we can help you, how we can get you started in the pipeline if you're not already onboarded to it, and how we can get things going for you. And we hope you attend the second part.
Alrighty. Thank you all for coming today. Oh,
looks like we have a question from Dr. Schwaber. Hi, Dr. Schwaber. Can you evolve automatic mapping? I think I can answer this. If I'm going for, we're continually updating our software and making improvements based on suggestions and feedback, and our automatic segmentation tools are always being improved. And you can go back to your data files at any time to improve the segmentation you've done already, or if you want to reanalyze it as well.
Alrighty.
All right.
We don't have any more questions and we are past our time, so we will just end the webinar now. Thank you again for attending.
Thanks, everybody.
Bye for now.